Cryptococcus is a fungus found in our environment where we live in all around the world. And it usually doesn't affect us as healthy individuals, but it can cause very severe meningitis in people whose immune system is weakened. For instance, people living with HIV and AIDS, they are prone to a lung infection with Cryptococcus, and it's very dangerous because the infection can spread from the lung to the brain, causing a fatal meningitis. I'm Greetje van de Velde. I'm a research professor at KU Leuven. That's the university in Leuven. That's a very nice town in the center of Belgium. And in my lab, we work on unraveling pathology and therapeutic options against infectious diseases.
I'm Jim Swoger. I'm the head of the mesoscopic imaging facility at EMBL Barcelona. I met Greetje when she came to the SHARP lab at the CRG while I was working there. Her expertise is in vivo imaging, so she can look at the samples live as the disease develops, but the resolution isn't as good as one might like.
I was looking for a methodology to fill the gap that we have between the information we get from in vivo imaging methods on the whole body level and information that we can get from histology on tissue sections with classical microscopy in 2 dimensions.
Most microscopes are designed to get very high resolution, but of fairly simple samples, like a 2D layer of cells on a cover slip. What we try to provide at the mesoscopic imaging facility are solutions that allow you to image larger samples, like mouse organs or developing embryos. The solutions we provide here allow you to look at them intact, so you get the full 3D picture of the sample.
We started a collaboration to check the feasibility of mesoscopic imaging of entire mouse organs that are infected, to really look with cellular resolution and in 3 dimensions what the spatial organization is of pathogens and also the host cells. And this collaboration is still going on now that they are at EMBL.
The main challenge that we had to overcome for this CryptoView project with Greetje was figuring out how to label the samples so that we could really see the details that we want to see. But in the end, we found that we could label the cryptococci cells so that we could see individual cells within the intact brains and lungs.
The protocols developed in collaboration with the MIF are not only applicable to my own research, but also to other disease models. And it's really nice to see that through the MIF, that this novel methodology becomes available to a much larger research community. What I truly appreciate personally about working with the MIF is that the staff of MIF is truly passionate about imaging, getting the best out of your samples, and it's very energizing to work together.
It's always a pleasure when one of the instruments that you've built is able to take pictures and show the biologists things they've never seen before. It's, wow, we didn't know it was doing that kind of thing.
The mesoscopic imaging data that we got through the collaboration with MIF really allowed us to do novel observations that were never seen before. We could observe that new Cryptococcus lesions are always organized around the microvasculature, and this gives us clues on possible entry mechanisms of the Cryptococcus in the brain. And also this is an important insight towards understanding the pathogenesis of the disease.